Hey everyone, welcome. So, welcome to the lecture. I am Aishman and I'm going to teach you today. So, first of all, I'm Aishman. I am from India and I am studying my medicine in Luigi Venvitli, Campania, which is situated in Italy. And this lecture is for IMAT. Okay, this is dedicated to the IMAT exam so that you can prepare according to it. The book here I'm using is the Cambridge A level. So, what are we going to take a motive of this lecture? So, first of all, this lecture is a trial that if I'll teach in what way I'll teach you guys, how I am going to teach you, how I am going to communicate with you guys. So, today's topic they are cell, then structure of cell, and different types of cells and their components. So, like cell, okay, cell, the basic unit of life, uh, which collectively forms a human being. And then after that, structure of cell. Then we will study structure of cell, then different types of cells like prokaryotic and eukaryotic cell. And then in eukaryotic, there are plant cells and animal cells. After that, we are going to study about different types of components in cells like ribosome, mitochondria, nucleus, and etc. So I think we should start, okay? So first of all, here we are using Cambridge book and it is written why cells, but I would say it should not be why, it should be how. Should be how cells are doing this, how are we alive, how a composition of atoms and molecules is working in such a fascinating way. Okay, so how are we going to study here? Because I'm going to teach like for an year maybe. So in that case, I'm going to teach you line by line of this Cambridge book. Okay. So first of all, a cell can be thought of as a bag in which chemistry of life is allowed to occur. So what is a cell? Cell. What is a cell? Cell is a round, maybe rectangular in any many different shape. It is just a closed bag in which there are different function occurrings like energy formation, like glucose formation and excretion, like many different things occur in this so that a organism can work. Like cell is the most basic unit of life, so in cell there should be a thin membrane around it which controls the exchange of material between cell and its environment, and the feature of controlling it leads to its property known as. So that what there's a property in cell which is called being its membrane selectively permeable. Okay, so in cell, when this is a membrane, right? So we all know about concentration difference and osmotic pressure, because of which things can go from here to here. And membrane are in chemistry, membrane are of three types: really permeable, and the second one is partially permeable. Okay, but what we need it to be selectively permeable, so that a cell can take whatever it needs. It do not depends upon just these two. So cell has the plasma membrane. What it does, it controls what molecules are going inside, what product is going inside, what is going outside of the cell. Okay. For cell biology, what we are going to do, we are going to study it in the next topics, but we are not going to study like microscopy and stuff because these are not required. Okay. Okay, so our topic is now about animal and plant cells, and we are going to learn about them by first learning about the common things in them and then differences between them. Okay, so we are going to use these diagrams as well. It is of plant cell, and this one here is a animal cell. Okay, so in animals and plant cells, each cell is surrounded by a very thin cell surface membrane, which is called the plasma membrane. Okay, many of the cell contents are colorless and transparent, so they need to be stained to be seen. Okay, and I'm telling this to you guys that in a cell, or like in a human being as well, like cells are transparent. Okay, you need staining, different sort of stainings to look at them. You need different colors to look at them, but a cell individually is a transparent cell. Okay, each cell has nucleus, which is a relatively large structure that stain intentionally and is therefore very conspicuous. So what do we mean by that? Okay, so here it stains intentionally means when we are doing staining, it is showing like very dark colors and it is taking too much of that stain material, and it is staining too much parts inside it. And what does it represents being intentionally stained? It represents that it is very detailed, it is very conspicuous, it has too much of material adjusted in different way in a very close area. Okay, the deeply staining material in the nucleus is called chromatin. So the material which is stained inside that nucleus, what is it called? It is called chromatin. Okay. Chromatin is a mass of loosely coiled threads. And what are these threads made of? DNA. The material collects together to form visible separate chromosomes during nuclear division. So what happens that this material during the nuclear division, what happens that chromatin condenses itself, DNA condenses itself to form what? To form chromosomes. Okay, it contains DNA, deoxyribonucleic acid, a molecule which contains the instruction that controls the activities of the cell. Okay, within this nucleus, an even more deeply stained area is visible, which is called the nucleolus. Like, let me check if we have a diagram here. No, we don't. It should be down here. Mm, yeah, here. So here we are looking at that. What is it? It is nucleus. This part is nucleus. Okay, but here at this region the staining is different, and it is called as nucleolus. Okay. Mm, yeah. A nucleolus is made of loops of DNA from several chromosomes. How it is made of? It is made of loops of DNA, but from several chromosomes. Like several chromosomes, DNA is looping around in this region. And also, what happens is in this region, the formation of ribosomal RNA. Okay, the number of nuclei available. Sorry, the number of nuclei are variable. Okay, like there's paramecium in which there are multiple number of nuclei. Okay, and 1 to 5 being common in a mammals. It is, I think, a very important thing, so I am highlighting this. Okay, it may be useful in further question, in it is maybe useful in IMAT, although this question haven't arrived until the exams I have checked. Okay, so maybe we will not need to remember it, but for knowledge purposes it is very important. Also, we don't know what examiners are going to give us in the upcoming exams, because we all know that the competition is increasing slowly. Okay, the material between the nucleus and the cell membrane is known as cytoplasm. So when we look at that diagram, here's this part is the nucleus, right? And just for context, just think that it is the cell membrane, the plasma membrane. Okay, so here this all of this part all of this, what is it? It is cytoplasm. Okay. And cytoplasm, what is cytoplasm? Cytoplasm is a aqueous solution in which there are different organelles sorry, like microvilli, like mitochondria, like lysozymes, like Golgi body. Okay. So here, cytoplasm is an aqueous material varying from a fluid to jelly-like consistency. And how does it varies? Because like if there's too much organelles in a single place, maybe there's a jelly-like substances are there. And if there are not too much of organelles on a single place, then maybe there's a watery fluid, there's a solution which contains different amount of substances. Okay. And what are organelles? Many small structure can be seen within it. So the small structure like lysozyme, like mitochondria, like Golgi bodies, they are called organelles. And organelles can be defined as a functionally and structurally distinct part of a cell. How can it be defined? It can be defined as a functionally and structurally distinct part of the cell. Now, what does it mean by functionally distinct part? Functionally distinct part means that each of those structure have different functions. Like lysosome, lysosome has a function of eliminating every like unwanted substance inside it by the help of those hydrolase enzymes present in it. And like structurally different, like if you look at endoplasmic reticulum, like is there any endoplasmic reticulum? Yeah, here it is. So that will playing it first. If you look at the endoplasmic reticulum, what do you see? You see these lines, right? But if you look at like mitochondria, like here, if you look at mitochondria, it is a sausage shape or a cylindrical shaped cell, right? Sorry, not cell, a organelle. So they have distinct function and distinct structure. Organelles themselves are surrounded by membranes so that their activities can be separated from surrounding cytoplasm. What this statement means? So if you look at the at a organelle, like here's the mitochondria. Let me show you it. So here's the mitochondria. You can see there's a inside content here, like this. This is the inside content. But if you look at this part, like if you look at that red part, like if you don't look at that sky blue part, there's a membrane. So each organelle has a membrane. What does it does? It protects the inside material from the cytoplasmic material. Why? Because as we know that each organelle has a different function. So for that different function, it requires different environment, different type of materials inside it. And the cytoplasm has all the materials it needs, but it has also the excess materials which it do not need. So this membrane manages what does it needs and what it don't. Okay. Organelles themselves are surrounded by the membranes, so their activities can be separated from the surrounding cytoplasm. This can be described as compartmentalization. The the function of these membranes which manages the environment inside the organelles and outside and keeping the substances separate is called as compartmentalization. Okay. Having separate compartments is essential for a structure as complex as an animal. So, what do we get by having separate compartments? So, in this case, like if we have separate compartments, all the naked materials or all the organelles are in a separate area and the work can be done in a very efficiently way. Okay. Since Each type of organelle has its own function. The cell is said to show division of labor. Well, if every organelle is doing separate function, it is what is it called? It is called division of labor. काम का बंटवारा. A sharing of work between different specialized organelles. The most numerous organelles seen with the light microscopes are usually mitochondria. So, what does it say? It means that a mitochondria can be seen by the help of a light microscope. Okay. Mitochondria are only just visible but films of living cells taken with the aid of a light microscope. They are specialized to carry out aerobic respiration. The use of special stains containing silver enabled the Golgi apparatus to be did for the first time. So there were special stains used with silver which enabled us to look at the Golgi body for the first time in 1898, which was done by Camillo Golgi. The Golgi apparatus is a part of complex internal sorting and distribution system. So what does it means? The Golgi body can do internal sorting. It can sort different materials, different substances inside the cell, and it can distribute it to different location, different organelles within the cell. Okay. It is also sometimes called Golgi body or Golgi complex. Now we are going to talk about differences between the animal and plant cells. The only structure commonly found in animal cells which is absent from the plant cell is a centriole. So centriole is the only structure which is only present in animal cell, not in plant cells. Although there are some exceptions, but for now we are going to consider that. Plant cells also differ from animal cells in possessing cell wall, large permanent vacuoles, and chloroplasts. So in plant cells we can form vacuoles or chloroplasts, but we can't find them in the animal cell. First, we are going to talk about centrioles. Centrioles. Under the light microscope, the centriole appears as a small structure close to the nucleus. Centrioles are discussed. Okay, we'll study them later as well. Okay. Cell wall and plasmodesmatous. Now it is going to be very interesting. Okay. With a light microscope, individual plant cells are more easily seen than animal cells. Why? Because plant cells are generally larger than the animal cell, right? They are surrounded by cell wall outside the cell surface membrane. Cell wall is present outside the cell surface. Okay? This is selectively rigid. Cell walls are rigid because it contains fibers of cellulose polysaccharide which strengthens the wall. It is a important line. What gives the cell wall its strength? Then the answer is going to be the cellulose fibers and the polysaccharide which strengthens the wall. The cell wall gives the cell a definite shape. Cell wall gives the cell a shape. Also, it prevents the cell from bursting when water enters by osmosis, allowing large pressure to develop inside the cell. Cell walls may also be reinforced with extracellulose or with hard material. In cell wall, there could we could found extracellulose or with a hard material called lignin. We can find lignin as well. With what does it gives the extracellulose and the lignin? It gives it the extra strength, which is sometimes generally needed in the region of the plants like in stems, right? Because stems are need to be stronger. Cell walls are freely permeable. They are freely permeable. On the other hand, plasma membranes are selectively permeable. And because they are freely permeable, they allow the free movement of molecules and ions through to the cell surface membrane. Plant cells are linked to neighboring cells by means of fine strands of cytoplasm called plasmodesmata. Let me tell this to you. Wait, is it present here? Ah, so when you look at the diagram, it is a diagram of a plant cell, and you look at here. Here it is the plasma membrane, and here it is the cell wall, right? And if you look at this region, there is a another cell wall, which is of different cell, enabling cell, right? And here, if you look, there is a presence of plasmodesmata. And what does plasmodesmata do? It connects two neighboring plant cells with each other so that the transfer of material between them, like the cytoplasms, can be done very easily. Okay, which passes through pore-like structure. Plasmodesmata, they are pore-like structure in their walls. Movement through the pores is thought to be controlled by structures of the pores. Movement through the pore is controlled by structure of the pores. Like if a pore is large in size, a large material can be passed through it. If a pore is smaller in size, only small materials can go through it, right? Now we are going to study about vacuoles. Vacuoles, although animal cells may possess small vacuoles such as phagocytic vacuoles, which are temporary structure. In the another hand, mature plants often possess a large permanent central vacuole, and these vacuoles they often occupy very large space in them. Like if you look at that, it is a vacuole and it is occupying a very large space, maybe like 40%. But these vacuoles they can even occupy 90% space of a plant cell. Okay, the plant vacuole is surrounded by membrane, the tonoclast, which controls and exchange between the vacuole and the cytoplasm. So what does plant vacuole do? It controls the sorry. So what does a tonoplast do? It controls the exchange of material between the vacuole and the cytoplasm. And what does a vacuole store inside it? It stores excess material in it generally, okay? Any type of excess material. The fluid in the vacuole is a solution of pigments like excess amount of pigments, excess amount of enzymes, sugars and other organic compounds including some waste product. It can also store waste products and also like mineral salts, oxygen, carbon dioxide. So anything in the plant cell which is in excess amount and it the functioning parts like chloroplast, mitochondria don't need it at that exact moment. It it gets sent to the vacuoles. And if we are looking at that and we are thinking, oh, the excess amount is going to it means it is managing the material going inside and outside of the cell. It means that it is a selectively permeable membrane, right? And being selectively permeable membrane, then we can also say that its working nature is same as that of plasma membrane. But both of those membrane like plasma and the tonoplast, their working function, their working nature, their mechanism is different from cell wall, because cell wall is a freely permeable membrane, right? Vacuole helps to regulate the osmotic pressure of the cell. That's a very good information to have. So vacuole, by keeping the products in the cell in a very regulated way, it also helps the osmotic properties of the cell to be managed. The flow of water inwards and the outwards, right? As well as having a wide range of other functions. For example, the pigments which color the petal of the certain flowers and the parts of some vegetables, such as red pigment of beetroots, may be located in vacuoles. What does it means? That when we look at a leaf, it is green in color. So its green pigment is stored where? It is stored in vacuole. Okay. Now we will study about chloroplast. In chloroplasts, they are found in green parts of the plants, mainly in the leaves. They are relatively large organelles, and they can also be seen by a light microscope. So, till now we found two organelles which can be seen by light microscope. What are those? What are those? These are mitochondria, and the second one being chloroplast. Okay, so they are found in green parts of the leaves and mainly green parts of the plants and mainly in the leaves. Okay, they are relatively large organelles and they can easily be seen by light microscope. It is even possible to see tiny cranes, or we call crana, which is a plural word, singular plenum, inside the chloroplast using a light microscope. These are the part of chloroplast that contain chlorophyll. Okay, we can find the screna inside a chloroplast, and they are also situated in like a pillar like shape, like this, like this, like this. Okay, and what does they do? They absorb light during the process of photosynthesis, the making of glucose inside a plant body, and which is the main function of chloroplast. Points to be note. First, you can think of a plant cell being very similar to an animal cell but with extra structure. And what are those extra structures? They are like vacuoles, like chloroplast. Okay, and they are generally larger than animal cell, because if they have extra structure inside them, then obviously they will be larger than a normal animal cell. Okay, do not confuse the cell wall with the cell surface. Remember, cell walls are relatively thick and physically strong. So there's a difference between plasma membrane and the cell wall. Okay, first of all, cell walls are stronger and thicker, and plasma membrane are very soft and very thin. Second, cell walls are freely permeable, but on the other hand, the plasma membrane, they are selectively permeable. So, okay, they are not partially permeable; they are only selectively permeable. And if you ask me what is the difference between partially and selectively permeable, so um, a partially permeable can be a membrane which lets some compounds go through it, but it do not let another type of compound go through it. But if we look at that, a partially permeable membrane can be the one which have like big holes and small holes. So if you look at a big hole membrane, it can let pass through the large molecules, right? But on the another case, if a membrane has only small molecules, it cannot let pass the large molecules, sorry, small holes, then it cannot let pass the large molecules through it. It is the partially permeable. Okay. But in case of a plasma membrane, it is selectively, it decides when does it needs the product it wants. Okay, when it do not needs the product, it has ability to open and close its gates, which are called channels. Okay, which are made of proteins. We will study about them in the upcoming chapters. Vacuoles, they are not confined to plant cells, animal cells. So vacuoles are not confined to plant cells. Hmm. What does it mean? Let's study the whole paragraph. Animal cells may have small vacuoles, such as phagocytic vacuoles, although these are not usually permanent structure. Okay, it means that vacuoles are not just limited to the plant cells. Animal cells may have vacuoles as well, like phagocytic vacuoles, like lyso, lysosome, which makes a phagocytic vacuole. So, what is it here happening that when we study about vacuoles, sometimes we have misunderstood that vacuoles are only present in plant cells, but it is wrong. They are also present in animal cells. Also, one more thing which I wanna add here is that um, plant cells have only one large vacuole. In case of animal cells, they can have multiple, but small vectors. Okay. Now here comes